Start off with the parent chain of 3 carbons. So now what we're going to do is we're actually going to start off with our double bond on our N carbon to the oxygen. So here, and by doing this, what we're doing is we're eventually going to create our carboxylic acid on the end. Double bonded O. So then we're going to add next. We're adding our other oxygen, and this is part of our hydroxyl. So we add our hydrogen along with our OH, and then so this part of the model so far is our COOH, so our carboxylic acid group. Then next we're going to do our nitrogen. So first we attach here. We have our nitrogen atom, which has the ability to make 3 bonds. So we have our N, and attach our 2 hydrogens. And then to finally end it off, we have our last carbon right here, which we are just going to create all of our hydrogen bonds. And finished alanine. And so here you have again, this is your carboxylic, you have your amine.
Okay, you ready?
Yes.
Okay, what's the name of it?
So mine is alanine, or
Good.
alanine as I sometimes say.
And it's a subgroup of what larger group of macromolecules?
So what do you mean? Oh, it's proteins. Okay.
And where's your R group?
So this is my R group right here, the methyl that has the 3 attached hydrogens.
Good. And as you can see it here, is this molecule polar or non-polar and why?
Non-polar. So
as we see it here.
Yeah, it's non-polar here. So and what I found is
why is it non-polar?
So these 2 atoms right here, this is polar. This is polar, but the main thing when looking at amino acids is its side chains. So this side chain here with the methyl itself is non-polar. So therefore.
So if I were to like cover up these 2 here, would it be polar or non-polar?
I believe this is non-polar.
Good. And why is it non-polar?
It's because it's not asymmetrical. It's like like symmetry as well.
Like around.
Yeah, like around the carbon.
That carbon there. Good. And what do we have existing between here and here?
Oh, this is hydrogen bonds.
Is there an equal sharing of the electron in between that bond?
Can't remember, to be completely honest.
Not sure.
No.
Do you know what it's called when we
It's
have
the
an
electronegativity.
Yep. Do you know what's formed when we have an unequal sharing?
I don't remember.
Not sure.
Okay. So so when placed in a solution, what ends up happening is the hydrogen from this carboxyl group then switches over to your amine in order to create NH3. So when in a solution with, say, our glycine, what ends up happening is the hydroxyl group from here pops off and combines with our hydrogen from our amine in order to create our water, therefore allowing our carbon and our amine, or nitrogen, to connect with each other, creating a dipeptide. And then this is a peptide bond in between these 2.